KBRIN's faculty-mentored Student Biomedical Research Program offers year-round part-time mentored research opportunities for students. KBRIN-supported faculty at Kentucky's regional colleges and universities provide research training and graduate school and career guidance. The program offers access to state-of-the-art equipment and procedures and opportunities to attend and present at state, national, and international meetings.
So I have a funded project from the National Institute of Alcohol Abuse and Alcoholism. We have laboratory-based research to look at whether people want to drink more when they drink alcohol with energy drinks. And then we are also looking at the implications for impaired driving. Everything about the project is student-driven.
So I'm part of the complete process, everything from developing a study, going through the IRB application and approval process, as well as recruiting, conducting intake sessions to do the screening, the complete conduction of the experiment, everything from the beginning to the presentation and hopefully potentially the publication of the research. The beauty of this lab is that our research is so applicable to a lot of people with alcohol use issues. It's all new and it's fascinating, and I couldn't be more fortunate to work here.
I had Dr. Marchinsky for our research methods and tools class. So going through this experience has really helped me put the principles that I learned in that class into real action. It's helped me see that research is the foundation for all of our knowledge, and it's really exciting when you get to discover something new. So I definitely think I will be continuing my study in research and hopefully pursuing a career in research as well.
I'm a cellular molecular genetic biology major with a chemistry minor. My mentor is Dr. Aaron Strom. So for this project, currently I'm trying to create a strain of yeast that is what's called a homozygous knockout for a specific gene and see how that affects the stability of the yeast genome. I used to be terrible at genetics. I didn't do very well in that class, so that's the reason I joined this lab, is because I wanted to get a better understanding of genetics as a whole, and cancer genetics was interesting to me, so I wanted to learn more about it. I have a much better hold on genetics as a concept, lots of different processes that I didn't understand before. I worked with some gel electrophoresis equipment, a tetride dissection microscope, and it's so cool. You can go in and pick apart microscopic cells. You can pull them apart and set them different places, and you can see them, and it's totally cool.
Our lab's research, we are primarily interested in the role of transcription factors and how they can drive the development of an organism, as well as later the function of an organism and its implications in disease. Primarily we're interested in development of the pancreas. My students actually drive most of the work we do. So I have currently 2 graduate students, and they're both working on different aspects of the project. My undergraduates also help out with both of the graduate student projects, and they're also looking at some of the more biochemical aspects using tissue cultures, and they then look at the proteomics and post-translational modifications of these proteins.
We're working on some mutated cell lines that Dr. Zaruth made, and we're looking at some of the expression of GLIS3 in the empty cell lines where they're lacking it, and when you add glucose to it, how the cells react. He's helped me a lot in discovering what I really want to research and what career path I want to choose and how I want to pursue my PhD. I think next summer I'll be attempting to get an internship at MIT over the summer.
My name is Nancy Ann Webster, and I hope to go to a PhD program as soon as I graduate from here to go and get my doctorate in pharmacy. This past summer I actually had an internship where I worked in a specialty pharmacy. I got to see the research here, and then I get to go and see how it's applied after.
I've been working on another project where we are actually going in and editing the genome of the model organism. We'll make a protein with GLIS3 attached to a protein that will actually glow green. So what that will allow us to do is look for this green glowing color throughout development, and that'll allow us to see when and where this protein is expressed.
KBRIN has funded me to look at modifications that are not found in yeast cells but are found in human cells. So we're hoping to discover some new enzymes that could be important for human health. I'm a new faculty here, and KBRIN actually gave me some startup money, so it helped me get my lab going, and then getting the KBRIN Idea Award allowed me to bring in several extra students this summer. I actually have 8 students in my lab right now.
Coming into the lab, I really didn't know a lot because I'd never done research before, so basically everything in here was new to me, and then I got working with it, and now I call myself the queen of making gels. Yeah.
Past few weeks I've been working on making in vitro transcription of a T7 gene so we can like knock out genes in the cells that we're needing. I've definitely learned a lot just about like genetics and biochemistry in general, because like I haven't gotten to that point in my studies yet, and it's always something I was really interested in. Actual DNA and RNA extraction and transformation. In vitro transcription is like a really cool process that I got to do like by myself here in the lab. So just like things like that you wouldn't do just in a normal class lab. Like I was always interested in biochemistry and genetics, but I had never really had full experience, but this really gave me that experience that like solidified what I wanted to do.
This experience so far has not changed my career plans, but it did offer me another backup plan because I realized that I actually enjoy being in the research lab, which is much different from being in your typical labs that you have to take with all of your courses. You're not graded on numbers, you're graded on, are you trying different things, are you coming up with different things to try?
So what we specifically study is what makes this germline cell a germline cell. To isolate those proteins or RNAs that are specifically localized to the germ cells and look at them biochemically. Most students that I support, they haven't done research at all. It has been amazing to watch how, in terms of, their scientific expertise has been developed from inexperienced person from from that stage to quite developed and sophisticated thinker, and that's a part of what makes me excited about supervising my undergraduate research students.
I've learned a lot of reproductive stuff about the flies since we dissect them all the time, and just along the way, Dr. Arkov always finds a way to throw in different information that might find useful in later classes or anything related to genetics.
My research project is about pyruvate kinase and its role in germ cells in Drosophila melanogaster systems. I engaged in a lot of different processes and also in using a lot of different equipment, and it improved my understanding in biotechnique.
In addition to faculty, students can receive training in laboratory procedures from graduate students.
I'm a bacteriologist. I study the responses of bacteria to DNA damage and how that relates to the bacteria's ability to mutate, become drug resistant, and study these processes in ways that can have application to human disease, such as cancer. One big important part of this second renewal grant was directly sponsored by KBRIN. It was derived from some investigations of genome-wide RNA expression in response to DNA damage, a very expensive type of experiment that KBRIN was able to fund and fund several of us investigators actually to do next generation sequencing. So that formed an important extension of the work I was already doing. I had not envisioned being able to do those sorts of experiments on the modest R15 budget, but with the assistance of KBRIN, then I was able to use that and kind of leverage that kind of information into my renewal, and I think it helped a lot.
What we are doing here is we are trying to grow graphene quantum dot to be used in human plasma to basically deactivate the pathogenic bacteria and the viruses inside the human blood. Students, they are actively involved in the, the experimental part. They do analysis part as well, and I'm expecting those students to, analyze the data as well. So right now they're basically doing, the characterization, executing the experiment, and, the helping with the technical stuff.
After producing graphene quantum dots, I do some purifications, and then we have to do some data analysis to be sure that we have graphene quantum dots. I didn't know about laser techniques. I didn't know about the characterization methods, but now I know them.
My job is to analyze these dots, look at them under an electron microscope, use a spectrofluorometer, things like that in order to get different spectrum and to see exactly what we have when we create the solution. So we do have to do a lot of supplemental reading and things like that in the literature in order to find out what we're looking for.
As far as this bacteria experiment is concerned, I've been fundamental in developing the prototype boards, the boards that are actually going to be irradiating the sample being blood, and we're applying quantum dots into them, and we're going to see what happens when we apply different wavelengths of light. And from there, we'll hopefully be able to get some great publicational data. I will hopefully be accepted into a medical physics program. It is a PhD program. I intend to do research in that field as far as medical imaging. From there, I just like doing research. I'm going to keep on doing it. Research and development has been my life for over about 10 years now, so it's not going to stop.
My project is using Xenopus laevis, which is the African clawed frog, as a model organism, and we're interested in studying vertebrate embryonic development. So the students do pretty much all the work. They definitely are involved in every aspect, so they help collect the embryos, they help manipulate them in different ways, and then we do a number of experiments to see what might be happening. So
mainly I work for Dr. Shifley on the immunohistochemistry side of things, but recently I've been kind of overlapping into some of her other students' work too, as we kind of all help each other out. We dye the embryos in antibodies, and then I go downstairs and I look at the fluorescence and see which antibodies are active and which aren't. And then in the lab itself, basically we all take turns making the solutions and autoclaving. I've taken both biology labs through NKU, and I think I've learned a lot more in the actual practice of research than I did when taking the labs themselves. It's just a lot more interactive, and I feel like when you're invested in a project, you get a lot more interested in the outcome of things and a lot more involved in how your results were affected by what you did in the lab itself.
Our project that's funded through KBRIN is a longitudinal study of the development of self-harm behaviors in adolescents. So we have been visiting 2 different middle schools and 2 different high schools in Kentucky, and we have surveyed the students there on a number of behaviors, including self-harm behavior, but also some other factors that might be related to that, and trying to see how they all develop over time. Yeah, the students actually do a lot of work. They manage a lot of our materials. We have our master list of participants that we're keeping track of at the different schools to make sure we're getting the same kids each time. And they've been going to the schools with me to help administer the surveys to the students. And then once we get back to the lab, they're doing a lot of data entry, because we have all these paper or pencil measures that have to be entered into our database.
Basically, I feel like it helps me to learn how to do research myself if I wanted to conduct a project. It just kind of puts it into a real-world application for me. I've always wanted to be a clinical psychologist, focusing on adolescents, specifically women. So it's kind of just furthered that for me.
KBRIN grants provide funds for equipment that grow Kentucky's biomedical research capacity, giving students exposure to tools such as this inverted microscope at Murray State University, the ChemiDoc imaging system at Eastern Kentucky University, or this HPLC, or High Performance Liquid Chromatography instrument at Eastern Kentucky University, the Potentiostat galvanostat sensor in Northern Kentucky University, sometimes an entire laboratory facility, such as the one at Morehead State University. KBRIN funds unique equipment that is very specific to a project. While very few U.S. institutions have a Peapod, it has been crucial to Rachel Tinius's research.
If we're just looking at birth weights in babies, we might be missing something. So body composition just offers more information about how much fat versus lean mass a baby has, how potentially mom's metabolism and mom's exercise levels during pregnancy, how those might then relate to body composition in their babies.
Education and training opportunities in bioinformatics are offered through the Kentucky INBRE program. The annual Bioinformatics Summit is a joint venture with the University of Tennessee. Researchers, educators, and students come to hear from leading specialists. It also provides unique opportunities for collaboration and forging links between members of various institutions. The KBRIN Next Generation Sequencing and Data Analysis Workshop provides training and skills that are central to the acquisition and analysis of NGS data. The workshop is held at the University of Kentucky and is open to faculty and students from private and regional colleges and universities. During the 3-day workshop, attendees acquire hands-on experience in NGS systems and use bioinformatics tools to analyze real-world NGS data. Functions such as the Kentucky Academy of Science annual meeting or posters at the Capitol help students with presentation skills and networking. KBRIN also offers traveling grants for national and international meetings.
It has opened a ton of different doors for me. Last year I went to the Southeast Regional Yeast meeting and I actually met a guy from the University of Georgia who works in the cancer genetics lab there, and he told me to apply for vet school there. If I get into vet school, I would be able to work with him in his cancer genetics lab and further my education as far as that goes, and so that's pretty exciting.
I'll be working kind of solo on this project, and I'll be presenting at Scholars Week and KAS next year.
So we did do also 2 different conferences this summer. With KBRIN grant, we went to the National IDEA Biomedical Research conference in Washington, D.C. And I actually won a travel grant for that, a student travel award. And then also we went to Wisconsin, Eau Claire, Wisconsin, to a smaller international conference for the International Society for the Study of Self-Injury.
Students' work can result in the opportunity to publish in peer-reviewed journals.
But right now, I believe we have enough data, and they're writing a manuscript now, so I'm expecting them to analyze the data scientifically.
So I'm working on a few individual projects. The first is my thesis, where I'm using curvilinear regression to look at non-suicidal self-injury frequency and its relation to outcome variables like suicidal ideation, depression, and anxiety to see if those relationships are in fact nonlinear.
And my project is the activation of bacteria by using graphene quantum dots synthesized by pulsed laser. This project is my kind of main project, and maybe this is gonna be my thesis.
Next semester I will be conducting my honors in psychology project in the lab, and at this point I believe my study will be looking at impulsivity and decision making under alcohol mixed with energy drink. So I'm really excited to take Dr. Marchinsky's research and kind of apply it to what I'm interested in, and kind of take the reins from what I've learned and apply that to my own project.
KBRIN is really often thinking about students and developing that biomedical workforce, and so many of my KBRIN funded students have either gone on to graduate school. I have several students who are about to finish PhDs in biomedical research areas, or they've gotten jobs immediately upon graduation in hospitals as part of clinical trials programs. And so whether a student wants to work right away and be part of the biomedical workforce immediately, or they want to do something more long-term like get a PhD in a biomedical area, KBRIN has allowed them to achieve those goals.
I have been recipient of KBRIN grants myself, and I'm confident that those grants have been instrumental for me to hire and support excellent undergraduate students who have gone to graduate schools as well as medical schools after they graduated from Murray State.
I plan on getting a PhD in human genetics.
I definitely want to continue with research, like go to grad school for this field.
I wanted to do to continue my career until I became a professor. So I was already going to do my PhD, my master's. It's just helping me to motivate even more, so that I can continue on, carry on.
Before starting research, I really hadn't considered it as a career, but now with as much experience as I have with it, I feel like I could really see myself going on to do something in biological or psychological research.
Now I think instead of more counseling, I'm more geared towards the research that's going to bolster all the prevention and intervention methods that are going to be happening.
So my eventual plan, I would like to become, to work in academia and to become a teacher and a professor and a researcher. And so that's my lifelong career goal, essentially.